viewing that. Let me go here. Okay. So I hope this is a bit clear, and I'll just move this here and I'll just open the slideshow so it's more clear so that you can see it. Okay. Okay, perfect. Okay, so for today's sort of webinar that we've sort of prepared for all the scientists today. So again, welcome if you're new to the sort of weekly exploration club. This session will mainly focus on SpatialX, or sort of how we can leverage spatial transcriptomics data, whether that's bulk resolution or single cell resolution. So particularly the case study for this week will focus on a very relevant clinical sort of challenge in the field, which is how we can investigate sort of glioblastoma infiltration and the sort of tumor core differences using the spatial transcriptomics data sets. And more importantly, how we can leverage spatial analysis within SpatialX, so very particular features, to really look at capabilities like region segmentation, correlation networks, differential gene expression, or even transcript localization in the image. So this is what sort of features we'll sort of try to cover today, really to uncover if there's any therapeutic targets relating to infiltration within sort of glioblastoma tumor core samples. Okay, so what is SpatialX? For anyone, again, who is new to these webinars and haven't used SpatialX before, you might have been a current user of BioTrain Lens, which was our former spatial imaging technology platform. So really, SpatialX enables the scientists to really analyze spatial data across multiple technology modalities. And this can range from bulk, single cell, and the subcellular resolution level. So really, this ability to be able to integrate multiple technologies and simultaneously look at transcript location, where in the sort of location of the mRNA transcript is it located in the tissue. And this is quite important when it comes to identifying therapeutic targets that can be used. Okay, so some of the challenges that we face within SpatialX or within other platforms and why we've sort of released this new system was really to face very important challenges. And some of these important challenges include the integration, again, of multiple technologies, being able to actually run cell type annotation. So this is quite important when you're working on bulk resolution spatial, when previously you would have had to run deconvolution methods, which were heavy, took a lot of time, and there were several methods that weren't really applicable depending on what tissue you were working on. And the other step from there was how can we take these annotations, how can we take all of these sort of spatial imaging information and run other downstream analysis that would be more advanced. So this would sort of allow us to really identify very rare cell populations where they're sort of located in terms of sort of the tumor sort of microenvironment or sort of adjacent to the healthy sort of normal tissue. So these were some of the challenges that we were sort of trying to really help researchers sort of solve. And this is why we sort of released this SpatialX system. Okay, so I guess to start with what we're going to focus on in terms of the main overall question. So this week was really to understand if we're able to utilize SpatialX and very specific features to characterize hypoxic, angiogenic, and infiltrated zones within glioblastoma. And that is within looking in different regions of the tumor core and how that can be different from other samples, which would be healthy sort of brain tissue that we can also sequence to see if there's any differences in terms of these molecular features. So that was really sort of the focus. Okay. And again, I'll sort of go into some background of sort of glioblastoma if there's anyone who don't really work on uro-oncology and they're new to this sort of field. So glioblastoma as sort of a neoplasm, it's a very aggressive brain malignancy by sort of looking or marked by its significant heterogeneity and poor prognosis. And this with a median survival of approximately 15 months, especially when they're sort of diagnosed. So that it's a really short time to really intervene and sort of look for therapeutic targets that can really help. So sort of GBM, or again, glioblastoma, can be characterized by sort of this necrosis and this microvascular proliferation, and also this sort of diffuse infiltration of the malignant cells surrounding the healthy tissue. So this is sort of what I've tried to show here, just an example of the different regions of glioblastoma and how these can be targeted as well, or analyzed further using spatial imaging data. Since this is really the technology that allows you to identify very specific transcripts and where they're located within sort of the tissue. So that's sort of the reason why we sort of focused on this sort of case study. And more importantly, when you're trying to analyze sort of these brain tissue, or particularly glioblastoma in the tumor core, these infiltrating cells are known to evade sort of surgical resection and drive tumor recurrence. So this is why typically through the year, you see a lot of patients not really sort of reaching after these 15 months, and there's no sort of therapeutic targets or treatments that have been really successful, especially when many sort of patients have different sort of mutations that might make the sort of tumor more aggressive. So I guess one other thing I wanted to sort of explain, just so you kind of get the idea when you're looking at the image and specifically the data set will show, is that there are very particular regions in sort of the glioblastoma tumor core. One which is just the tumor core as its whole. So this is where you would normally find sort of necrosis, you would normally find hypoxia, mesenchymal sort of like state cells. And this sort of hypoxic zones really drive this angiogenesis, leading to this microvascular sort of proliferation and this immune suppression. You also have other regions which are also quite important when it comes to targeting glioblastoma, which is the infiltration zone. And this is really coupled by malignant cells that can extend in sort of the peritumoral sort of brain region. And this can also sort of interact with other sort of cell types like neurons, astrocytes, and oligodendrocytes. So this is also quite an important local sort of area in the tumor that we'll sort of try to focus on today. Okay. And this is just an example of these different zones and where sort of they're located in the brain. So another really important part as well is really understanding how this sort of tumor progresses and how this specific type of glioblastoma can be targeted when it comes to looking at specific therapeutic marker genes or other sort of transcript information in the image. So particularly glioblastoma involves a range of molecular features when it comes to sort of progression, and it ranges from tumor regions, malignant cells, and that leverages pathways that can be adapted, or they can invade, or they can interact with the microenvironment. So some previous studies that sort of go into this area have identified very particular pathways that are involved within this glioblastoma progression. And this sort of involves notch signaling, signatic genes, angiogenesis, hypoxia genes, and these sort of mesenchymal sort of like state markers as well, which are more for invasion and immune modulation. Okay. So I guess looking into how we can utilize SpatialX, one of the studies that we prepared for today was this Xenium human brain data set that can actually be found in SpatialX. And for anyone who's not really sure what SpatialX is and where you can access it from the Biotrain ecosystem as a whole, this is where you can identify, you can access SpatialX right here, which you'll notice it's in beta version. So that just means we're constantly developing new features in the system, and we're sort of happy to get feedback when it comes to analyzing different types of imaging data. So when you're accessing SpatialX, this is the sort of interface of the platform. You'll see that you can create studies which allow you to import internal spatial imaging data into the system. So that would be if you're actually generating your own images. And the other side of the analysis, which would be this sort of public studies. So this would mean that you can search for particular data sets that are accessible in the system, and you would be able to then analyze that data further. So currently we're just having some updates being done. So unfortunately, I won't be able to show that part of the analysis, but I do have all of the sort of features that you can visualize in the data as well, just so you get an idea what functionalities you can use and what data output you can get from this analysis, especially if you're working on sort of neuroimmuno-oncology. Okay. So this is where you can find the public studies, and then you can explore these data sets further. So this particular study, so you get an idea how these technologies differ, which ones might be more accessible or more specific to your type of research question. So particularly the case study for glioblastoma, it utilizes the Xenium or the Xenium platform, and it mainly looks at in situ gene expression analysis. So this is data coming from adult human cortical sections, and they employ very robust gene expression panels that are designed to really capture the transcriptional landscapes of the image. And this might range from healthy, cancerous, or neurodegenerative diseases as well. So particularly, we'll be focusing on the healthy brain section, which is the adult human brain cortical sections, and also sort of the glioblastoma tumor core sample as well, which sort of encompasses the sort of identification of neuronal, endothelial, astrocyte, microglia, and oligodendrocyte cells, as well as other immune cell types that interact with the microenvironment. So this is sort of a study that is quite important because it has a lot of data that you can use, especially if you're just trying to explore how you would be able to use the spatial technology for your specific case study or research question before doing any of the sequencing. Okay, so this part of the live demo, I've sort of prepared already some results just because we're having some updates in the system. So some of the first questions that we really wanted to identify was how can we perform this gene transcript localization across the entire image, especially when you're looking at multiple images in the system. So for anyone who hasn't worked in this field and you're not sure exactly what that means, like what is transcript location in the image, so this particular method really looks at single cell spatial data, or it's used in single cell spatial data, and it allows to find sort of the transcript of the gene sort of alias or the gene target. And it enables you to really pinpoint in the exact spatial sort of locations or individual mRNA sort of transcripts within the tissue. So you're able to see all the counts of that particular gene and where it's localized within sort of the morphology of the image. So one of, I guess, the main questions was to really identify other new sort of novel targets that have been sort of used in evidence in other cancer neuron interaction publications. And this was particularly looking at APH1A, which has been shown to be quite important in NOS signaling pathways, ANGPT1, which has been shown to sort of have some effects in angiogenesis, CYNPR, which is more for sigmatic activity within the microenvironment, and other sort of marker genes like CD68, which has also been shown for more sort of immune interaction with sort of tumor core cancer cells compared with the sort of immune cells in those infiltration regions. So these were very particular targets I wanted to really explore in this study to see how we can gather information from this gene transcript localization feature. Okay, and this is just a bit of information why these sort of markers are quite important, especially if you're trying to really label regions of the tumor and you want to run differential gene expression or actually target very specific locations as well. Okay. So some examples here on how you can really interact with SpatialX, especially if you're opening multiple images. So if you're looking at sort of healthy donor brain cortical regions compared to sort of the glioblastoma tumor core. So this is actually the glioblastoma tumor core, and this is how you would normally see the image in SpatialX. And sort of how can we gather insights in terms of not only the transcript location, but what cell types or proportions can we see and where are they concentrated in the image? So in terms of glioblastoma in the tumor core, just by looking at the image or just exploring the image, we can see that there's a vast majority of cell types that are very specific to the tumor microenvironment. And in the case of sort of glioblastoma, we can see that there's sort of an abundance or an intensity of these endothelial cell sort of areas or regions. So if I zoom in into the image, you can see that there's sort of these regions where there's an abundance of these endothelial cells. And there's some other regions where you see this as well within the tumor core. Other cell types that are quite important as well that we're just able to see just by looking at the image is microglial cells. So we can also see an abundance in very specific regions, for example, here at the edge of the tumor core, and also sort of close to the endothelial cell. So this might mean that there's some sort of form of cell-to-cell interaction within the tumor core itself. And again, you can just visualize just from looking at the image that there's sort of a huge intensity of oligodendrocytes as well in the tumor, in very specific regions here as well, and also some astrocytes that are communicating with other immune cells here at sort of the right-hand corner. So these are things that you can gather just from localizing or just visualizing the cell type proportions in the sort of single cell resolution. And this is quite hard to see just from here, but these individual transcripts, these are just showing the number of transcripts that are located within the cell. So it's not very beautiful as you would see in SpatialX, but it just gives you an idea what you would visualize from the data. Okay. And as opposed, if we look at this sort of healthy brain region, cortical regions, we can see that they don't really concentrate on, there's not any abundance of very particular cell types. In contrast, we see that there's more sort of neuronal cells that are sort of communicating, but there's more sort of neuron cells compared to what you would see with sort of microglial, endothelial, and astrocytes. So these are sort of the main differences we can gather just from looking at the image and interacting with the different cell type annotations and where they're located in the image as well. And SpatialX, one thing I'll mention, when you open up the image, you are able to really filter exactly what settings you want to add into the image. So that's quite important. If you have images where you have immunofluorescence staining, you can also sort of increase or decrease the intensity of those stains. And you can see more where they're located in the image as well with the sort of cell to, segmentation model as well. So this is just an example, but there's a lot more settings that you can run in SpatialX. Okay, so how can we really identify, okay, how are these markers sort of enabling very specific sort of infiltration zones, and what insights can we gather as well from the results? So some of the results when you ran transcript localization, which unfortunately we're not able to really show today, when you input the genes or the targets that you want to really see where they're located in the image, you'll be able to actually perform transcript localization. So this will create sort of another visualization image that really tracks every number where you see this transcript located and in different areas in sort of the tumor or in healthy donor cortical regions. So some main results that we were able to find if later on you go into the study, which was that this Notch signaling gene is really localized in very particular infiltration zones. So it really maintains the idea that glioma plasticity really enables immune evasion, and this can drive an adaptability change or therapy resistant if you're sort of looking for immune therapeutic targets. So some examples on sort of SpatialX that we can show here, which is sort of the visualizations that you can generate from transcript localization, is sort of this comparison between healthy brain sample and then the glioblastoma tumor core. So this is what I wanted to show, is that this particular Notch signaling marker, APH1A, really shows that sort of they're infiltrated and there's a higher enrichment or abundance within very specific infiltration zones of the tumor. And this can be correlated with the cell proportions as well. So you can actually get an idea how many transcripts are actually located within specific regions for those cell types. So this is sort of an example of how you can use this feature. And again, there's several sort of analysis that we wanted to really understand. So other markers that are more related to angiogenesis, how can we sort of analyze this in the tumor core compared to healthy donor cortical regions. So we can also see that ANGPT1 is really enriched, particularly in endothelial cells. So we see more of an abundance where we saw very particular intensity of these endothelial cells really located in the tumor core. And this reflects sort of hypoxia-induced angiogenesis. So this might mean that it's a really sort of hypoxic zone, which supports tumor survival, especially for nutrient deprived conditions like the tumor core of glioblastoma, which makes it really challenging for any treatments to be successful. So again, if we see this looking at more of the localization regions, we can see again, compared to the healthy brain sample, we're able to see very sort of enrichment of where we had these sort of endothelial cells and astrocytes sort of communicating. So this is how you can gather some information. You would get the counts. So the counts of how many transcripts for that particular gene are sort of located in the image, and it'll give you more information as well at what sort of scale in the image you find that sort of localization. Okay, Apart from other results that we also were trying to really answer was if there was any involvement with somatic genes within the tumor core. So we can also see that there's a high expression with the infiltrated tumor regions and adjacent to the neurons. And this might indicate that there's some somatic mechanisms within the tumor that spread and sort of have this neural integration. So this is also quite an important or a potential therapeutic target that can be used for disrupting sort of these neural integration and invasion in the tumor core. And again, just to show you really the difference between healthy donor samples, what you would see based on the somatic genes, we can see that in the glioblastoma core, we have this sort of enrichment in the infiltration within sort of different regions of the tumor. So again, this is sort of some information really important when you're trying to really see what type of therapeutic target would be specific for this type of sort of tumor or this type of sort of challenge as well. And I think the last part is really understanding, okay, from here, how can I really understand if there's any immune interaction with other sort of neuronal cells or other cells that you would see in sort of the glioblastoma tumor core? So there's are some specific markers like CD68, which we also tried to test within the study. And we were able to see that it did sort of drastically elevate with other core markers that were more specific to microglia and astrocytes. And these are more pro-inflammatory markers that are invasive to the microenvironment. And this really correlated with CD68 because it also correlates the activation of microglia cells that are involved within these tumor-associated immune responses. So again, if you really want to run this analysis, you can also create this transcript localization, and you can see that there are sort of intensity or specific regions where we see sort of this abundance of this specific transcript target. And again, this is where we were able to see the microglial with the endothelial cells that are sort of interacting with the sort of immune response or immune dysregulation system. Okay, so I guess one other thing that I wanted to mention is how else can we really visualize this data? Because it might be hard to look at every point or every region, especially if you're not really used to analyzing spatial imaging data. So SpatialX also does include the ability to create many visualizations as well. And that is quite important, especially if you just want to validate that you're actually seeing the results and you want to see if that correlates to the actual gene expression count across different cell types. So just by creating these bubble heat maps, you can also create violin charts or other sort of type of hierarchical heat maps if that's more suited for your type of data. But just from this analysis, I can validate sort of what we were seeing in these transcript location images, which was that they're very particular targets that might be involved in synaptic activity, that might be involved in this immune crosstalk interaction or other sort of hypoxic specific markers. So you can see that CD68, for example, that is more of an immune microenvironment sort of interaction marker. We can see its intensity mainly within microglial cell proportions in terms of the gene expression of that transcript. And this is, again, we can also confirm that the somatic activity is very specific to glutaminergic cells, whereas we don't really see that in healthy sort of cortical regions in healthy sort of brain tissue. So we see it more intensity within glioblastoma. And we also see these other hypoxic markers that also have more expression than what you would see in the healthy donor cortical regions as well. So this is quite important, again, when you're really trying to understand how do these transcripts relate to the sort of location of the tumor core and how can these be targeted, especially if you're looking at different cell type populations in the image. Okay. So again, I wanted to also cover, okay, how can we take this sort of results that we've gathered from just looking at transcript localization, looking at cell type proportions within these localized region, how can we understand, okay, is there any correlational networks with specific spatial variable genes that can be targeted and specifically that are involved in specific pathways that are enriched within sort of the structure of the tumor core? So the second question of the webinar was really how can spatial segmentation identify very distinct tumor regions for therapeutic targeting? And this is by sort of using the region segmentation model within SpatialX, which is a new feature that you can actually access on your own study or on a public data set as well. So just from running this specific functionality, I did want to sort of specifically go into what sort of region segmentation is, because there might be, again, researchers that haven't used this model before. So this specifically is useful for spatial data because it really identifies spatially distinct regions that might contribute to some understanding or biological processes underlying these regions. So it's more of a method that leverages the co-localization of these genes. So again, going back to the transcript localization, and it looks at across spatial coordinates that delineate specific regions and specific pathways that are enriched within these regions. So it would give you more information that you can gather, okay, what pathways are involved within these regions as well. So some results that we can gather, and again, I apologize, we couldn't really show this live within the SpatialX platform, but when you run spatial segmentation, especially in the context of glioblastoma, we can reveal that there are very particular key molecular differences between glioblastoma and healthy brain cortical regions. And particularly, we were able to identify very specific genes that I've shown here in some of the results, which include BX1, which is a stress response marker, CLC17A7, which is more of a metabolic marker, and also PTEN, which is a tumor suppression marker. So this sort of markers really help us understand how this glioblastoma progresses and how we can highlight druggable pathways that can be sort of targeted. And again, normally you would get a result, an example here, where I can go into the specific segments that are the output when you run region segmentation. And each segment is very specific to the pathways that are enriched and also the particular targets within those regions that have a really high AU cell sort of enrichment score. So that's another feature that allows you to identify active sort of cells for that particular gene set or that pathway sort of gene set. And again, this is an example where we can see this within the glioblastoma tumor. So I wanted to show an example how you're able to see the different segments after running region segmentation, and then how you're able to also identify what particular cells and where they're located in the tumor have this higher enrichment of this gene set. So you can go through each particular target to see where they're mainly enriched, and if that correlates with some cell proportions that you also have. And you can also sort of look at the pathways that are also enriched within that segment. So some of the results here, which is a second one, is that the second sort of result that we were able to find is that particularly in infiltrative zones showing sort of overlapping genes. So both in the healthy donor cortical regions compared to the glioblastoma tumor core, we were able to identify that segment 0 had this shared sort of list of markers, ENC1 and NPTXR. So these are particular markers that indicate neuron tumor interactions and might be potential targets that both disrupt infiltration while also preserving normal tissue. So we were able to really see them interacting in both images. And this is just an example where I wanted to show that sort of these sort of markers are shared across these different tumor regions. And you can also look at the pathways that are specific to that gene set as well within these different regions of the tumor. So this is, again, some results that you can gather from SpatialX. And I guess some of the summaries that we can take from the overall aim that we tried to cover, which is really to understand these hypoxic, angiogenic, and infiltrated zones in the glioblastoma tumor core. So we were able to reveal, just by utilizing SpatialX and transcript localization, that there's particular distinct molecular features across hypoxic, angiogenic, and these infiltrated zones. And this was shown through sort of the enrichment of pathways like Notch signaling, which particularly we looked at APH1A, which is a specific marker for that pathway, angiogenesis, specific markers like ANGPT1, which was enriched in particular areas of the tumor core, and also the involvement of synaptic genes like C1, CYNPR, which is more of a sort of immune marker, and CD68, which really dominated very particular infiltrative regions. And this highlighted this adaptability of the tumor and invasion and immune evasion as well. So this is sort of the main aim. The second part, which was more looking at how can we take this further and run other methods like spatial variable gene analysis and other methodology relating to region segmentation, was how we were able to demonstrate how glioblastoma heterogeneity really has an effect on how you can really look at treatment response, especially if you're sort of trying to analyze samples from these patients. So we were able to identify very core specific pathways that drive metabolic reprogramming and angiogenesis. And this was specific to the tumor core, whereas we were also able to find shared marker genes that were known to be expressed within infiltrative zones that leverage this neuronal interaction and immune modulation as well within the healthy sort of brain tissue as well. So these were sort of the two main aims that I really wanted to cover. And again, if anyone has any other questions, something that I did want to mention, since this platform is in beta version, that means that we'll constantly be updating new features. We'll be gathering feedback from scientists that are actively using SpatialX for their own data as well. So just keep in mind, if you have any particular bottlenecks that you're facing, if the analysis is not enough and you need more support in other types of methods that need to be incorporated in the platform that are very specific to your type of data or technology, just reach out to the support team. So the support@bioterrain.com email really connects us to the engineering team or any of us from the CSM team as well. That can help you really gather that feedback, and then we'll sort of give that to the engineers that would be developing these other advanced features that are sort of upcoming as well. So you'll see here some are already accessible in SpatialX, which has the blue check. Some of them are still in development, and some like region pathway analysis, deconvolution, trajectory inference. Really, we need your feedback if that's something that would be very useful for the type of data that you're working in, so that we then sort of develop it further in the next coming months. So again, this is sort of the roadmap of SpatialX. And again, I just wanted to remind anyone who has joined the call today, especially if you're sort of on the Asia side, happy Lunar Year. So we won't be holding the sort of Exploration Club the following week, and we'll resume on February 7. So I hope to see everyone who joined today, because we always try to do new case studies that are more relevant to the type of work that users that have used Bioterrain have really sort of given feedback for us to sort of prepare. So thank you. I hope this case study was useful, and I'm sorry and I apologize again, we weren't able to really show on the demo on the live system. It's just being updated. So we hope that there's new features that you can use. So just reach out if you want to sort of use the platform and you want to try sort of these data sets to run your own analysis. So thank you, everyone. Okay, one thing I wanted to mention before we log off, there was a poll that was sent. So if you can just add your feedback to the poll, that would be great. That way we can know if there's any questions that were remaining from this specific case study. Thank you, everyone.